Oh yeah, you're on like the break.
But.
I'm so good that I got a concussion.
Queen of Egypt, also the Queen of Balling.
Mike. We got a good one. Where's
No one left.
I gotta be the first to tell you.
You want to go in, Mike?
Yeah.
Balling. She's been up here for a couple years old. Oh, wait.
All right.
You know, right?
All right. So I have something for you to work on. Have a seat. Okay.
Addie, you're so lost.
What happened?
Nothing.
Just set it there for now. You guys aren't working. Just set it right there. Okay, here you go. There's something she wants you to do, and I gotta clean up. So you gotta work on this now. You're welcome. It was gonna be on a trip, so she wanted a little bit. All right. Well, this stuff I have to move on. Oh, you know, I always need help. I'm over it. Thank you. Grab something. Bring it. What else you got? Let me give you your stuff.
Yeah, you're right.
Here you go.
This is straight. Yeah, we went into that table like that. Dude, you must see.
Are we just friends?
I remember English.
And I'll start cleaning up while you guys are working.
This one?
Blue raspberry massive.
And the last one's in it?
And the other two fatty acids.
This one's all right?
Yeah, I'm just trying to.
So, wait, that's not.
No, they're both fatty acids.
So this one's triglyceride?
Where I'm like, can't do that.
And then if the byproduct is like CH2O.
These are all saturated.
Sorry.
No, you don't have to be.
I'm here.
This young man likes to build up. I told him to sit here because he's uncomfortable sitting on the stool. And he goes through contractions with them. Right there.
Okay, that sounds good. Okay.
Here's the extra paper.
Is someone missing now?
She was
Probably wasn't the nurse because her heart rate is too high.
Her what? Her heart rate's too high? Okay. Thank you. Oh, yeah.
I didn't get to the, because I'm slow, I didn't get to the, I'm gonna go over this, I'm by myself after school today, so can I just leave that there, is that gonna be in your way?
No problem. They didn't get to
They didn't do
the
it because I was,
explaining things in the beginning, or?
yeah, too much, that's always my downfall, but I didn't have my, seriously, I didn't feel so well prepared because I had this, the club and then, I knew I had lead, but I didn't have my paperwork ready. But I was too tired. I'm just. I'm not asking you to. I'm going to sit there for school. There's no book that
Can I show you real quick before I leave?
I was going to work on it after school.
Come here, we'll go in the back.
Yeah.
And like with her
There's really nothing to it. I mean, you know, we did it the other day together when she was.
Oh, she said.
Yes.
Is it bottom part right?
Yeah, it got cut off. They should be all hydrogen. Sorry. So on the first, like on the top, like fatty acid, like the H's are all cut off because I like covered it with other things. Is this what I wanted to do today? I think.
Try and then bliss, like bliss, or yeah, try and go for it. LYC. I drew up. Alright, so I've done this. What molecules? Well, I don't even know what molecules are. I know that this one is going to be a.
Don't look at the. Don't look at the slide.
This is unsaturated, right?
What is the new model?
Basically just puzzle.
I think you're confusing yourself. Oh yeah. Something about table pencil.
Carbon per hour. 1, 2, 3, 4, 5, 6, 7, 8.
Saturated. Omega what? Omega what? What is omega what?
Oh, omega.
Omega what?
No, it's not that they're back on. My hand is strong, and I
Oh my God, strong. That's like for me saying I'm too strong because I hit the water fountain.
And then these are and then what is it? What's it gonna? How much should be in there? I would have done circle what's in a half.
You're still be running a little
You can kind of draw it in on the thing itself if you want. Let me see. Well, you don't have to draw, like redraw the whole thing. Yeah.
So you're just planning
But that's
to shut out all
you're not right about that. No, the ester bond is not just an that's an ether.
Tell me how that works.
And you don't have to know that, but that's not an ester. An ester, like just doing this, like this, and if this were like like this, if I did this, that's not the right thing. Why?
Why do you have to do that?
This is what you kind of did, Arsh, right?
Because I don't have my the oxygen does not have anything else on it. I'm not.
Right. So
Yeah, I need to
the
double
thing that
bond
makes it
it.
an ester is that it has a
Carboxyl, yeah.
It has to have a carbonyl here. So it's like not only this thing itself that makes it the ester bond, but it's that it's also connected next
Yeah,
to
yeah.
other, because that used to be the carboxyl group.
Honestly, I was
Yeah.
using it at home. I might have worked on mine, but
That was just going to be 3 times.
All right, can I have all your attention? Are you ready? Did you get an answer? Somewhat? Yeah? Is this pretty easy? I hope so. All right. So what's this called? It's better than what? Carbohydrates?
Yeah.
Well, once you get through carbohydrates, then it like gets easier because you like if we'd done this first, then you would have said carbohydrates.
tragic.
All right. What is this? Can someone tell me? This is bliss triglycerol, right? All right. So I added two. I gave you two of these, and each of these is identical, right? So what are those?
Saturated fats or saturated?
They're saturated. You can call them carboxylic acid, or they're fatty acids, right? And what makes it a fatty acid? What are the two parts that make it a fatty acid?
The
Adam?
double bond to the oxygen and the bond to the OH.
The double bond, the two things that make it a fatty acid. So there are two parts. So you're right, but that's only one part that you're talking about. So this double bond right here, and then the OH here, which is called what kind of group? What functional group is that called?
Carboxyl.
Carboxyl,
And then
right?
It's also a carbon chain.
Not just carbon, but what else is on it? Hydrocarbon chain, right? So that whole thing, so this whole thing makes it a fatty acid, right? Okay, this is a fatty acid, so they're both saturated fatty acids because there are no double bonds, right? All right, what about the last one? Have fun. Be careful. All right. What about that last one, George?
Omega-6 unsaturated fatty acids.
Good. 1, 2, 3, 4, 5, 6. Oh, so that makes it omega-6, unsaturated, because anything that's omega or anything that's like cis or trans means that it's unsaturated. So by definition, it's So you can't have cis for a saturated fat, right? Because there's no double bond. So like, which side would we be talking about, right? All right. So once you say something that's omega, and once you say something that's cis or trans, you know immediately you're talking only about unsaturated fats, right? Fatty acids. All right. So. Yeah. So this is unsaturated cis omega-6 fatty acid, right? Okay. So now what was the job that you guys had to do? You had to combine them together, and what did we make by doing that? We were making a triglyceride, right? First, and what was the process called? Dehydration synthesis, right? You guys are like experts at this by now, right? So H and OH, right? They come together. Two hydroxyl groups interact to make water, right? Do you see the water? That's the drip here, right? And then we have the ester bond, which is here. But you have to know that an ester bond always has this carboxyl group next to it, right? So don't just make this a regular like CH. It has to be those two O's are there, right? And then that's dehydration synthesis. Any questions? No? Okay, so a triglyceride in animals is known as what?
Saturated.
Okay, so it's saturated, but it's and what state is it? What state is it? It's solid, so it's just known as a fat, right? Okay. All right. Now that I grab my double chip. All right. And then when it's in when it's in a plant though, like plant products that have oils, it's what state of matter is it? Liquid, right? And then it's made from plants, right? And so it's an oil, right?
Yeah.
Are you guys all right here? Yeah. Except for and then fish oils are those are omega 3 and 6. Okay. Are you guys okay for this today? Yeah? Okay. All right. So today we're going to continue our discussion of types of lipids, right? And we're going to do all the rest of the types of lipids, okay? So now that we've established that this is what again? This is a triglyceride, right? Hannah?
Why are the numbers like not?
Because I changed the slide. But it should be 1, 2, 3, 4, 5, 6. Okay? Is that weird?
Yeah, I just got confused.
All right, I meant to erase this. Okay. So this is what do we say this is again? You guys should immediately look at this and be like, I know what that is. What is that? Mary, what is this? Okay, Mary. It's a triglyceride, right? That's what she was going to say anyway. All right. So now I want to, so I'm going to talk about, in order, we're going to talk about 3 different, and I showed you a picture the other day, 3 different lipids that make up the membrane, right? Do you guys remember what they were called?
Glycolipids and the
All right. So what's the basic one that like kind of makes up the whole like substance of it? Those are the just phospholipids, right? That's what makes this bilayer here, right? So these are phospholipids. And then what do we say a lipid is called when it has like a glycan on it? This is a glycolipid, right? And then another lipid that we're going to talk about that's in the membrane is that 4-membered ring one. called? Does anyone know what that's called?
Oh, shoot. Forgot about it. Wait. hold
It's
on.
actually just called cholesterol. Okay?
Oh, cool.
So these are the three lipids that are in the membrane. Right?
Amazing.
Are you guys with me?
I hope so.
All right. So to make a phospholipids are kind of similar to triglycerides. So we're going to start with the triglyceride, right? What you do is you take out one fatty acid, right? So let's say we're going to take out this one, right? All right, boom, it's gone. Good? Easy enough so far? Okay.
How to make phospholipids?
Yeah, how to make phospholipids. It's like a recipe. Okay, so now what do we have? Now we have, this thing is it's basically, it's a triglyceride minus the one fatty acid, so it's kind of like a diglyceride, you could say, right? And then instead of, why did I not, okay. I don't know why I did that. All right. So we take out the we take out the third fatty acid, and then what we do is we replace it with a phosphate group, and a phosphate group looks like this, right? So it has like P in the middle, and then it has all these O's around it. Do you think that? What do you guys know about O's? O's are what? They're very electronegative. So that means that they'll what? What will they like to do? They'll like to attract electrons, right? So will it be hydrophilic, or hydrophobic? Arsh?
They would be hydrophilic.
Hydrophilic, because water is charged, so phosphate group is charged, so it's going to be hydrophilic, right? So what you do is basically this is all you do. Like a phospholipid is just like a triglyceride. Instead of one of the fatty acids, there's a phosphate group there, right? But the thing that's a little different about it is that the phosphate group doesn't want to be near these things that are like hydrophobic. No. So basically what happens is it just sort of rotates to the opposite side. So instead of coming off of this side, it ends up just coming off of this side. So it kind of makes a longer molecule, right? So here's the animation. I don't know if it's going to work. We'll see. All right, so that's going to go over there. And that goes over there. That took me like a really, really long time. I'm not proud of that, but so the idea, you get the idea, right? So basically, there's a phosphate group over on this side, and then the tails are on the other side, right?
Is that an R at the end D?
Yeah. So R just basically distinguishes between different types of phospholipids. You don't have to know the difference between different phospholipids, but know that there are different types, and they would be distinguished by what this R group. Yeah.
But doesn't the oxygen on the top have an extra electron, or is that just...
What oxygen on the top? The one up here?
Yeah.
This one right here?
Yeah.
Yeah, well, so that's why it's like considered negatively charged. So there are like actual like net charges on it, and they're negative. All right, so yeah, just... So that's what a phospholipid looks like. We're good? Okay. Do you guys have you copied that down kind of, or? You don't have to copy down every single carbon.
Is that O on the bottom, like underneath the P, is that double bonded to?
Yeah. So phosphorus is in like a different like period on the periodic table, and it can actually hold bonds with like more than 4 other members. So it can have more than just 8 electrons. That's something you don't have to know. Okay. So this is a phospholipid. If I gave you a picture of this on a test, you'd be like, that is a phospholipid. Really easy, right? And you'd be able to distinguish that from a triglyceride, right? But even though they are really, really similar. Okay? Or if I gave you a triglyceride and I said, make a phospholipid, you know how to do that, right? Okay. I'm going to turn it around sideways, all right? So, and it's not the same exact phospholipid, but I'm just going to show you a different view of a different phospholipid. And you guys have to recognize which area is which of it. So where's the phosphate group? Arsh?
It's bonded to that carbon over there, third carbon.
Yeah, it's way at the top where the phosphorus atom is, right? Yeah. All right, what's that thing? What's that next row? Omar, what's that next thing right here? This 3 carbon guy right here. What's that?
That would be the 2 fatty acids.
Two fatty acids are down here.
Yeah.
But I want to know what this thing is right here.
Glycerol.
Good. Glycerol, right? That's the glycerol part. And then those two things coming down are the row.
Saturated and unsaturated.
Good. There's a saturated fatty acid, right? And then there's a unsaturated fatty acid, right? Okay. Easy? Okay. So the top part, the phosphate group, is hydrophilic, we said, because oxygen really wants electrons and it's negatively charged. So the phosphate group is considered the head part, and that's hydrophilic, right? And sometimes instead of actually depicting it like in the chemical structure, they'll just put like a circle. And that's why those circles there in my diagram over here, these represent the what? The phosphate head, right? Okay. And then basically the whole rest of the molecule, right? Like this, the tail part, those are considered the hydrophobic tails, and a lot of times those are represented by these guys right here, right? Hydrocarbon tails. Right? And so what do you notice about this molecule? It has one part that's what? Hydrophilic, and one part that's hydrophobic, right? So we call this type of molecule that has like two different regions, like one that's hydrophilic and one hydrophobic, we call it amphipathic, right? Which means like both. So it has like both components, right?
So
Arsh?
is the glycerol, is that part of the tail or is it part of the head?
It doesn't really matter. I'd say if anything, it's part of the. You know, they never really like incorporate that into like the. They're really just skipping over it.
The amphipathic, that's a molecule that.
Amphipathic is just like the adjective to describe any molecule that has both a hydrophilic part and a hydrophobic part. Sure?
So do you have to have like one saturated, one unsaturated?
In typically in phospholipids there is one that's typically it does look like this, right? But it doesn't necessarily have to be. They could both be totally saturated or they could both be unsaturated. But usually they're like this, right? Okay, good question. All right, are we good so far? Okay, so there are three different like shapes. If I just like take a bunch of like phospholipids and I like throw them in water, what's gonna happen? What would they form? And there are three different options. So you could be right if you said any of them. Okay, they could form a ring, right? Why? Because what likes the water? The head part, right? So would they be facing out or would they be facing in?
Out to keep the tails.
Yes. So, and that's just something called a micelle, right? So essentially it's like a little ball, but then they all orient themselves so that the heads are facing out and the tails are facing in. That's one option of how this thing could, what the phospholipids can do. Depending on different conditions, and we're not going to get into like the specifics of that, but this is one option and that's what it looks like. It's one layer of phospholipids, right? You see, there's only one. And then it's just the tails and the inside, right? It could also form, just kidding. It could also form what's known as a bilayer, which is basically like this, right? Which means that they're all, again, here's water, but these protect themselves by putting their tails in so the tails don't get exposed to the water, which they hate, right? And the phosphate heads just remain on the outside, enjoying the water, right? So that's the second type of structure that could be formed when phospholipids are put into water, okay? And then there's one last option, right? And this thing is called a liposome. And it looks like this. So basically what's happening here? There's like a bilayer kind of. But so there's in the inside there's a little what's on the inside of the liposome then? There's like a little section of water inside, right? And then it forms like this like kind of bilayer in a sphere around that little watery inside, and then there's water on the on the way outside too, right? So this actually is an artificially created molecule. So people manufacture these, right? Usually it's artificial. And suppose I have a drug that I want to administer to a patient, right? I could put it in what kind of form do you guys usually take drugs? Pills, right? So suppose I give you like a pill. Usually like you have like those capsules. What are capsules? What's that stuff on the outside of capsules made of? Like gelatin or something that's like pretty, that dissolves like pretty easily, right? But suppose I have this drug that I don't want to dissolve like so easily, right? I can put it in a liposome, right? And if I put it inside of here, if it's a hydrophilic drug, then I can put it inside of here, and then it can go through the body kind of protected until it reaches its area where we want to administer it, right? Or if it's a hydrophobic drug, I can put it in this portion here, right? So it can also be transported by that liposome, right? So this is, it's like a growing technology. This is like a method or a way of like administering drugs, right? Which is just one of the applications of it.
Would it make sense to describe them as like almost like a hollow ball or something like that?
It's kind of like, but it's not hollow because it has stuff in it, right?
But like that hollow part's filled with like water.
And if you think about it, our cells are actually like huge versions of this. So like cells, obviously the inside part is like way bigger.
Cytoplasm, sort of water.
But this would be the cytoplasm on the inside, and it's basically like that's basically like a mini type of cell, but it's called like a vesicle, right? Yeah.
But how does like the drug know where to go? You know?
Well, so there are like other markers that you can put on the outside of it. So like, and that's where like oligosaccharides and things like that come into play. So like, if you want it to like target a certain type of cells in your body, you can like put a marker on it, so it will like know to go there. It's like amazing that we can do that, but yeah, you can mark it.
What's the word for like the like when it has a hydrophilic head and a hydrophobic tail?
Amphipathic. So amphi means like all around, right? Like an amphitheater, right? And path means like sort of, you know, if you're sympathy, it's like feeling kind of. So it like feels everything, meaning it has like both hydrophilic and hydrophobic properties.
Wow.
Good, right? All right, so we're good with phospholipids, right? Their main job. What's their main job? To phospholipids. So not liposomes, but phospholipids in general, like these little guys, like this thing right here, that one thing, that's a phospholipid. What's the main purpose?
Wow.
Cell membrane.
Cell membrane. It makes up our cell membrane, right? That's like the main purpose. Okay. So the second thing in the cell membrane that we are going to talk about is glycolipids, but we've already talked about them, so we're not dwelling on this. So a glycolipid is a lipid attached to a sugar, a carbohydrate, and what level of carbohydrate? An oligosaccharide, right? And what is that oligosaccharide also known as? If it's a sugar portion of a molecule, it's called the glycan, right? So the glycan is this purple thing up here, right? Here's the glycan. And then the bottom part is just the lipid portion. There's no fancy word for, it's not like the lipan or something. It's just the lipid portion. Okay. All right, where do you think these are coming off? Do you think they're coming off the outside part of the membrane or the inside part of the membrane? Outside, right? Why? Because what's their main job? Cell recognition or signaling, right? So it wouldn't make sense to like signal inside of your cell, right? It really wants things outside of the cell to recognize them, right? So they're on the outside of the surface of the cell membrane, and they're used in recognition or signaling. Okay, we're good there? I don't mean to go too fast, but I don't want to dwell on this because you guys already knew this, right?
What is 11 oxygenate cleaner? Is this just water?
No, I think it has some sort of like cleansing properties to get. I think actually, do not spray it on people's faces. Thank you. But yeah, it's.
Why does it have such a like fancy name? I was reading it the other
No idea. And it's, but it's not like an official, like that's just like a label that someone taped on it. So it's not like a.
Hydrogen peroxide, I know what that means now. It's at 1 hydrogen per oxygen, so it's like H2O2.
All right. Moving on. All right. So we said So we said we know what phospholipids are, right? We know what glycolipids are, and what is this four-member thing again called?
Steroids.
They're steroids, but it's called cholesterol, right? So cholesterol, the main thing that cholesterol does is it helps the membrane stability. What does that mean? All right. It helps it stay stable or consistent, right? And it's kind of paradoxical because it's the same molecule, but at different temperatures it does an opposite role, right? For example, at high temperatures, what type of like consistency would a membrane be? If it's really hot, what do things do? They kind of melt and they get all like loose and like kind of fluid, right? Is it too good? Is it good for the membrane that like is like the the structure of our cells? Is it loose? Is it good for it to get like all loose and like crazy and like fall apart? No. So what would I do if this membrane is starting to get melty and like fall apart with all these spaces in between and it's going to let all this other stuff flow through? What should we put in those spaces to keep it stiff and firm?
Cold.
You put in a bulky molecule like cholesterol, and then it actually adds substance to the membrane. So at high temperatures, so cholesterol adds substance, and will it increase the fluidity of the membrane or decrease the fluidity of the membrane? Will it make it more fluid or make it more stiff? It will make it more stiff, so it will decrease the fluid nature of it, right?
So the what's called the phospholipid would keep the melting thing.
Yeah, they'd be kind of like melted at that temperature maybe, but cholesterol kind of by fitting in there and like filling in those gaps because it's such a bulky molecule, it can like kind of help the membrane maintain its shape at high temperatures. So it's like a support thing at that level, right? All right, good. Okay, tell me about at low temperatures. What happens at low temperatures? Things like to do what?
Like they freeze would be like too stiff.
Oh, whoops. Sorry. All right, so at low temperatures, they get really stiff and they're like really packed together, right? Things want to pack together. Like think of like things that are solid, like saturated like fatty acids. They're really solid and they're stiff and straight, right? So at low temperatures, they're like that, right? Do we want the membrane to be stiff and frozen? No. So what do we do to prevent it from being too stiff and frozen? We add cholesterol inside, right?
Steroids.
So at low temperatures, what is cholesterol doing here? Is it making it more fluid or less fluid? More fluid. It's making it more fluid, right? So do you see the kind of like contradiction? At high temperatures, it makes it less fluid by being like acting as a sturdy, like stiff. Are you okay?
Yeah. She wants to see me back again at 2:20.
Okay. So at high temperatures, it actually adds bulk to it, right? It makes it stiff. And at low temperatures, it is keeping them spread out so it doesn't get too stiff, right? So it prevents the tails from packing too closely, and then it increases the fluidity at that level, right? So do you see how it's contradictory? Yeah. All right. Are we okay? Okay. Good? Yeah? Okay. So let's talk about like other things about the membrane fluidity just for curiosity's sake, right? So what effect will all these things have on the membrane fluidity thing? So increased temperature, what will it make the membrane fluidity more fluid or less fluid? More fluid. Why do you think so?
Higher temperatures, so there's like more activity.
Yeah, so higher temperatures melt things, right? So it makes them move around faster and then it makes them more fluid, more liquid, right? Okay, so think of butter, right? If you melt butter, it gets liquid because it's high temperature, right? Okay, think about the length of the hydrocarbon tails on the fatty acids. So suppose I have a hydrocarbon tail that looks like this, right? Versus a hydrocarbon tail that looks like this, right?
Maybe more of a solid.
So these guys, look at the long ones. They have all this distance to do what with each other? To pack together, right? To intermingle in their hydrophobic interactions, and they're going to pack together, right? So which one's going to be more fluid? So the long length, is that going to increase the fluidity or decrease the fluidity? It's going to decrease the fluidity because they're going to be like all tangled up with each other and stiff, right? So that's decreasing the fluidity of the membrane, right? All right, so what about this? If I have right, so what about the high degree of saturation? If it's really, really saturated, meaning what? Is it straight or bent? Straight, right? If it's really saturated, is that going to increase the fluidity or decrease the fluidity? It's also going to decrease the fluidity, right? So having more bent or unsaturated tails is going to increase the fluidity, right? And then what do we say about cholesterol again? It's at high temperatures, what does cholesterol do?
It makes it more structured.
It makes it more structured, or it decreases the fluidity, right? And then cholesterol at low temperatures, it spaces them out, right, so that the tails don't pack too close together, and so it actually increases the fluidity at low temperatures. Okay? This all makes sense, right? Yeah? Is it too... No? Yeah?
Only like the contradictory part of the cholesterol makes it weird.
This one's a little weird, but this makes sense, right?
Yeah.
Because they're straight. This makes sense because they're long and like interacting over a long space. That makes sense because melting versus freezing, right? Okay. All right. So on the test, if I gave you something like this, you'd be able to do it, right?
Yeah. Cool.
All right. What's this thing right here? Cholesterol, right? What about this guy right here? That whole green thing, the whole green thing itself is a glycolipid, right? But then what about this top part over there is the glycan, right? The bottom part is the lipid portion, but I don't actually have a line for that.
The tails or the
yeah, what is this whole thing? What's this thing? That's a phospholipid, right? The whole thing is a phospholipid. And then what are these? Hydrophobic hydrocarbon tails, right? And then what are the red balls here? Hydrophilic phosphate heads.
Those are phosphorus.
Okay. Easy?
Can you go back to the last slide so I can see?
This one?
Yeah, that one. Thank you.
Lidu?
Would the glycan also be the oligosaccharide, right?
The glycan is, yes, it is an oligosaccharide. But yeah, good? Okay. All right, so cholesterol, one of the jobs then, cholesterol is what? Good, so it's maintain the stability of the membrane because at either extreme temperature, it causes it to do the opposite and maintain its homeostasis, right? So that's one job of cholesterol, which is a steroid, right? Let's talk about the other steroids, and you can recognize them again because there are 4 fused rings together, right? They're not rings like in the way that we had, remember in our, what's it called? In our carbohydrates, remember they look like this? Do you guys remember carbohydrates, right?
Yeah.
So they look like this, right? Are those fused together?
No, they're bonded.
They're bonded together, but they're not fused. Fused implies that they like have something where they're actually sharing like some sort of side here, right? Right, so they're actually fused together, right? So
How is that natural or no?
yeah, it's just a molecule. It's like a molecule that rearranges itself by like grabbing onto another part of the same molecule, right? Okay, and so besides like cholesterol, which is for membrane stability, the other steroids are mainly used for signaling, right? So you don't have to copy these down, like the chemical structures, but I just want you to appreciate that they all consist of 4 fused rings. Do you notice that? Yeah? And I want you to look at, like, so it starts with cholesterol up there, right? And they're all these other ones are all derived from cholesterol. So is cholesterol bad for us?
Isn't it
In
bad?
fair amounts.
Well, so like we need cholesterol, right? So just to say like unequivocally that like cholesterol is bad is wrong, right? Because obviously we do need it, but there are some times when cholesterol can get problematic. But that's not this cholesterol. We'll talk about that in a second. So the way we actually use the word cholesterol is wrong all the time. It's like inaccurate. Okay? So anyway, so cholesterol serves as membrane stability, but it also serves as the precursor. You guys know what a precursor is?
Yes.
It's like the first, like the grandfather sort of, right? Of these other ones. So all these other ones, progesterone, have you heard of that?
Yes.
Testosterone, cortisol, aldosterone,
Yes.
estradiol, those are all derived from cholesterol, right?
We don't need to know this, right?
What?
We don't need to like know.
So you don't have to like, I'm not going to be like, draw estradiol.
Cool.
But you have to recognize, if I gave you these, you have to be able to like be like, those are the steroids.
I see.
Right? If you say that's a phospholipid or if you say that's a triglyceride, then I'm going to be upset. But if you say it's a steroid, and that's fine. All right? All right, just remember it looks kind of like a little house over here, and then these 4 little, hexagons. I mean, yeah. All right. And I'm going to go through each of these in a second. Shh. Okay. Don't copy them. I'm going to give you the names right now. All right. So cholesterol is the precursor. Shh. All right. So progesterone. Have you guys heard of that?
No.
So
It's a combination of estrogen and testosterone.
Sort of. It's a different hormone though. It's totally different, but it's really important in pregnancy, right? And sexual development. All right? So progesterone is pretty important. Aldosterone is also, it's important in your kidneys, right? In regulating your levels of like salt, your osmotic levels, like water, right? So progesterone is like in like pregnancy and sexual development. Aldosterone is for maintaining water levels, right? Testosterone is for the development of male characteristics. You guys know that already. Estrogen or estradiol, which it's derived from, is for female characteristics, right? And then cortisol is a stress hormone. Have you heard of that? Right? So when people get stressed out, they release cortisol, right? Which is not good.
Cortisol increases testosterone.
What?
Is cortisol the antiinflammatory or is that
Cortisone? Are you thinking? Cortisone is also like a steroid cream, right? Like when you have like an itch or something, you can like rub a steroid cream on. Yeah. Okay.
Stress rashes?
What?
Stress rashes?
Stress rashes, cortisol?
Yeah.
It just involved the whole stress response, right?
No, like the cream you were talking about. Like stress rashes.
It's used for any sort of like, I mean, if you have a bug bite, you use that. Yeah. Okay. All right. So basically, but what's the point? What's the purpose of all these? What do hormones do?
Homeostasis. I don't know.
They can regulate homeostasis in some ways, but the main point is just signaling.
Oh.
They're signaling like between cells. Do you guys know the difference between a neurotransmitter and a hormone?
Yes. Oh, the neurotransmitters, they react to stimuli like immediately, and then hormones like they develop over time and then they change like over a long period of time.
Okay, all right. There's a more direct that's true in many cases, but that's not, because sometimes the same exact like chemical can act as a neurotransmitter in one context and then a hormone in another context. Do you know, Lita?
I want to say like neurotransmitters what gives information to the brain, and like hormone is what your brain releases to.
Not exactly, because so for example, there are certain chemicals that act both as a neurotransmitter and as a hormone, right? It's defined by the target cell. So in a neurotransmitter, one neuron shoots out a neurotransmitter, and the other one takes the very adjacent cell responds to it, right? Whereas a hormone gets sent somewhere else through the body to a target organ, but it doesn't like go right to the cell right next to it, right? So in any case, it's like the purpose is like a hormone, you send signals to some other organ in your body to produce some sort of response. Okay? So we're good on steroids. Easy? Okay. So you'd be able to recognize the 4, and you know that cholesterol is a steroid, but cholesterol is also involved in membrane stability. Right? But it's also the precursor of all the other steroid hormones. Okay? All right. And you guys are going to do projects on each of these, so that's why I'm not going to go into them in too much detail. But you guys are going to research one of these and then present. So we'll learn much more about what these do. That's why I'm not going into too much detail. All right? All right. Okay. So how do cholesterol and triglycerides get all around our body, do you think?
Lipoprotein.
Yes.
Okay. Well, suppose we just... That's a really good point. Why? Do you think that they... Well, so are they hydrophilic or hydrophobic? For the most part, if we had to classify them as one, we'd say they're hydrophobic, right? So can we just put them in the blood and like send them along their way, on their way?
No, there's like water all over.
Yeah, there's water all over in blood. They don't want to deal with it, right? So what's going to happen? Lipoproteins, right, are going to carry them. Lipoproteins are going to carry them throughout the body, right? Cholesterol and triglycerides. Can you predict what the arrangement of a lipoprotein might have to be in order to be able to travel nicely in water, but also to guard this hydrophobic stuff on the inside?
I'd be like a circle, like hydrophilic things on the outside, and then like the hydrophobic stuff is like in the inside being carried around.
Exactly. That's similar to what? Can you think of what the 3 structures that we talked about? Which one?
The liposome. Oh wait, no, the micelles.
Yeah, it's very similar to the micelle, right? Okay. So if you look here, right, and because of the name lipo means fat, right, and then protein is protein, right? So you'll have lipo. I think some people say lipo actually. No, this is like lipo. Like liposuction, right? Like lipo. I've heard people were saying like liposomes. So I don't know.
Sounds like hippo.
2 P's, right? Oh, hippos are. Okay, that's a good way. Whatever helps you. All right. So if it's a lipoprotein, it has to be composed of lipids and proteins, right? So these are all phospholipids, right? And they're oriented with their phosphate heads on the outside because those are the things that are hydrophilic, right? And on the inside of the lipoprotein, what do we see inside of there? Triglycerides and cholesterols, and then we have on the outside are proteins, right? Do you think the proteins are hydrophobic or hydrophilic if they're on the outside? They must be to a certain degree hydrophilic, but maybe the inside of the protein could be what also? Hydrophobic, maybe. So what would the overall protein molecule be described as if it has a hydrophilic portion and a hydrophobic portion? Amphipathic. Excellent. Good. Okay. So if I asked you to draw a diagram of this, would you be able to do it? On the outside, you would draw phospholipids and proteins. Phospholipids oriented with their heads facing outside. All right. And then in the interior, which is hydrophobic, we would put the hydrophobic stuff like triglycerides and cholesterol.
I should probably describe that.
You couldn't draw it like this? I know that, like, kind of the way they cut it open, it's like harder to.
Yeah, like how do you use it?
There you go.
Oh, so
I could do a demonstration.
I vote she does. I can't draw.
okay. All right. This is actually the last thing. Are you guys ready?
So could you do a demonstration?
Huh? A demonstration of how to draw it? Yeah. You can.
Wait, what are we doing?
Do you want to? Okay.
Yeah, I can't draw.
Do you want different colors?
This pivot.
No, it's okay.
What are we doing?
I'm gonna collect your labs and then you're gonna do the worksheet.
I'm annoyed at him.
There's a worksheet, yeah.
Or can we just start our project on all the hormones?
I'll give you the instructions for it, but I think I was gonna let you do it tomorrow in class.
All of them?
I am looking.
Is that hard?
That looks different from yours.
Oh, that's so.
Oh, I thought that was gonna be the outside part. Didn't no?
I thought I could like be
Big brain.
dramatic. Like I just can't draw.
Damn.
It's like a 3D.
Language.
Also up there.
Doesn't exist to me, so I don't.
So
Whoa,
bad. I
hold
would have
on.
never thought of that.
Me neither.
I would have drawn like a circle and then erased it.
Same as.
Well, that's insanely.
Bro.
That's insane in the membrane.
Insane memory.
That's beautiful.
I'm so serious, like, good job.
All right.
Look, I still bad sometimes. How was mine so bad?
Puppies are you eating? It's cold, bro.
She's just been sleeping to the lead. Like every day.
Protein. Yo, protein, bro.
Oh, is that why she wanted to record?
Yeah, that's why she
was
Okay, so all right. This is the part where what I was talking about before. All right. So, LDL and HDL. Remember how I said before that cholesterol, in our colloquial use of it, if I say, "Oh yeah, you have high cholesterol," that doesn't mean cholesterol in the sense of like that 4-membered ring. It actually means you have high lipoprotein that's carrying cholesterol. Right? So our definition of cholesterol is actually a lipoprotein. We're not talking about cholesterol, the steroid. We're talking about cholesterol, meaning like a lipoprotein that's carrying cholesterol. Right? I'll elaborate on this in a second. So there are 2 different types of lipoproteins that carry cholesterol. There's one that's called LDL, which refers to the density of the molecule, right? So something that has like low density, would it be lighter or heavier, you think? Lighter. And would it have more proteins or more lipids in it? More lipids, right? Okay. But this one, so it's kind of lighter. It's not as dense, right? And basically all lipoproteins carry cholesterol, right? But it depends on where they're carrying it. So LDL, low density lipoprotein, carries cholesterol from the liver to all the body cells. So it's going like outward, right? And then when it's traveling outward, and because it is so not dense, what can it get if it's going out toward the cells? It's gonna start. It can build up in certain like arteries, and that can cause clotting or like these like it can cause like blockage, right? And that's why this is known as the bad cholesterol, right? So again, it has more cholesterol than protein, so it's less dense, right? And it can lead to buildup in our vessels, right? Which is not good, and that's why it's called the bad one, right? So what do you think, if this one's low density lipoprotein, what do you think the other one is?
High density.
High density lipoprotein, right? All right, and this one's the good one. That's why it's in green. And then, so it's actually carrying, where do you think it's carrying the cholesterol then from? From the cells back to the liver, right? And the liver then removes the excess cholesterol, right? Which could potentially cause like blockages or, fatty buildups. So that's why it's a good thing, right? So it's like removing it from your system, right? And then these have more protein, so they're like denser. They have like more of a ratio of proteins to lipids or to cholesterol than the other way around, right? Yes, George.
So the ester linkage, is that named after a person, or is that named after the 5 letters in cholesterol?
Very good observation. I never would have thought of that. I don't know the answer, but I think the name Esther, like you're thinking of like the woman's name Esther, or you thinking of like a.
Well, I was thinking like, I don't know.
I think usually Esther has like an H in it. I think it's S.
E-S-T-H-E-R.
That's Uncle Lester, I thought. But you were the Addams family for not the Addams family, but for Halloween, right? Wednesday, yeah.
I know.
This judge called me Monday.
Uncle, no, it's not Lester. Wait, I thought it's Fester. Fester, right?
Yeah.
Fester, not Lester. Okay.
Yeah,
What's the main guy? What's the main guy's? Oh, the main uncle? That's Uncle Fester.
As in the day?
no, because I was Wednesday for Halloween, so the whole day she called me Monday. Because I don't know. She's this
The
judge.
Addams family's too much.
Okay.
Let's do the tricky one.
Okay. We're done. Are you guys good? Wait. Not yet. Yes. Wait. All right. So. All right. I'm gonna listen to me, please. Shh. I'm gonna give you both the worksheet today, right? And that's for homework for tonight. And then I'm gonna give you the description for the project, right? You guys, I want you to work in groups of 2 or 3, all right? And it's gonna be another sort of presentation. But you guys know how that works, right? Like if 10 groups just get up there and just kind of talk about stuff that's just rambling on and on, right? Try to make it interesting. Try to make it try to be creative a little bit, right? I mean, I do want some slides to show that you for us to kind of to guide us. What are my main requirements for slides though? I don't want what? All right.
Big words.
No full sentences. I don't want people to I don't want a big line. And when I say no full sentences, I really mean it this time. No subjects, no verbs. They should all be fragments. If you can say it as a full sentence, if it sounds like a full idea, then
It's a caveman talking.
you should sound like a cave. Yeah, it should sound like a caveman, and you should try to minimize. You should minimize the text on it overall, right? So your job, what do you think it's going to be? You have to research not a steroid. It can be anything. It can be any lipid, but it has to be a lipid in the human body, right? And then presumably it will have some sort of role, right? And then I also want to know if that lipid is implicated in any sort of disease. So if there's too much of that lipid, is there a problem? If there's too little of that lipid, is there a problem? If there's something maybe it's made wrong, then there's a problem, right? Have you guys ever seen the movie Lorenzo's Oil? No? You'll watch it next year in anatomy. But it's basically this disease where this kid has his body can't break down long chains of lipids. So that's why he it's a very sad story. Adrenoleukodystrophy, I think it's called. I'm gonna tell you. All right, listen. So 15 points, right? Content, appearance, presentation. I'll come around and take a look at your slides, right? But I do want you to just think about ways that news, the way you guys did the news program, something where you can involve the audience, right? If you guys can involve the audience, or maybe do a case study, so it's a little more personal than just being this hormone does blah 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 blah, right? Try to make it think about ways to make it interesting. I will give you points for creativity. So all right, so I'm going to pass this out. I think you guys by the end of today, I think you should do some preliminary research, and I'll give you all day tomorrow in class to work with your groups. One thing I don't want to see when I'm walking around and watching you work in your groups, I don't want to see people just sitting on opposite sides of the table and just typing and you you do this part, I do this part. Because then you never have to learn about the other part, and they never have to learn about your part, right? So I want you guys to be talking to each other, and kind of talking about the stuff, right? That's the point of group work, right? So yeah, and then you guys will the things that you guys talk about in your presentations you'll be responsible for too. So find a way to make it clear and interesting so that your classmates can remember it better, right?
Are we presenting this on Monday?
You'll present it on Monday, right?
And then test. So it's after we can present and then
And then I think the test will be on Tuesday.
Yay!
Right? I think the presentations, I think we should stick to about 5 minutes for those, right?
We can post the test Wednesday.
No, that's so.
Postpone the test on
Wednesday.
next Wednesday. Morning, right?
Yeah.
Okay. All right, so listen, so by the end of today, you guys can't do the same lipid. And no one can do triglycerides because that's too easy. So you can choose a phospholipid that you guys want to do. You can choose a steroid hormone. You can choose cholesterol, HDL, LDL. You can choose a type of liposome that people are developing to administer drugs in patients, something like that, right? But it should be, there are certain lipids that are vitamins. right? But just do some research, and I'm gonna make a Google Doc, and I want you guys to all make sure your group signs up for that lipid, so that we don't have like a lot of people doing the same thing. And like, once someone does like a type of phospholipid, I don't want every single group to do a different type of phospholipid. That would be so boring, right? So think about ways to kind of like make it diverse, make it interesting.
I don't know who's my partner's gonna be because I don't have any friends in this class.
No, you guys.
Arnav Patel. Arya.
Can you guys be responsible enough to wait? Maybe I should wait. I'm gonna let's say you can work with anyone who you haven't worked with yet in the group, like for a project. So you can't work with anyone who you worked with before.
No, wait. No, bro.
Omar.
You guys can figure it out.
Alright. Yeah, so you're just gonna buy them? No.
What is this? The human capital trade? Hi.
I'll work with
Omar. Yeah, who else is in it? Wait, Arya and Arnav were in this.
I didn't work with I
got a smart group, guys.
You wanna work with
Who are you working with? I got Charles. What? Yeah.
me? Wait, is it part of two?
Wait, is it 2 or 3?
2 or 3.
No, we good.
I need a group.
Charles. But
Make sure that no one is excluded, guys. Be aware of who's. Yeah, and you guys, can you roll out the computer from out there?
Are they there? We write on the board what we call.
I'm gonna put on the I'm making a document, like a Google Doc.
By the way, I just wanted to let you know now, I'm going to email you because I was hoping that maybe I could meet with you someday at lunch before the test to go over what I missed while I was in the nurse.
Oh, yeah.
Yeah, because it seems like I missed a lot.
Yeah, unplug it and then plug it back in. What time did you come in? I don't remember when you came in.
I came in when you started like the lipoprotein.
Oh yeah, you missed a lot.
Right around there. Yeah.
All right.
I came in like the second, third to last slide.
Okay.
I was in the nurse for like the first half an hour of class.
All right. So when do you want to come in? Sorry, I need to make this Google Doc, but tomorrow.
Yeah.
Tomorrow is Friday. You're coming, Mike? You're coming tomorrow at 10:30, did we say? Can you come at 10:30 tomorrow? Is that better for you or is 11 better? Okay.
11's better for me too because I have a yearbook meeting at 10:30.
Okay.
But I could, like, if you need to.
No, 11 is fine. Yeah, come at 11, but you'll be with him, and we can maybe all go over it together. And I also need to talk to him about his classes.
Sounds good.
Can we do testosterone?
Yes, I need to make a. Hang on a second.
Oh, then we can just put it on the doc then. That's good.
Yeah.
On the gawk.
Lipid. I'm not sure.
Omar and I. Oh, yeah, my Are you ready in line to request?
Who wants to?
We're doing testosterone.
No.
What are you doing?
Testosterone. I like testosterone.
I guess you're not.
Really?
Dang. That hurts.
Whoever can get their.
Miss Pittman, it's the skinny ones.
The cute ones.
Oh.
The skinny ones.
Isn't always those?
I don't know.
Yeah. Oh no, maybe that was time we didn't.
fat.
Those are nice, right?
Yeah,
Are those not the fat ones?
no, these aren't the fat ones.
Oh, that's nice.
I never realized how large the computer laptops are.
One, it's like but it's only like 11.6 inches.
I
think look at these have the 15-inch because they're huge. I have the 13.
All right, go ahead, you guys. Oh, I have to share it with you, right?
Oh shoot! No, lock it.
Wait! No, don't run! Wait a minute. I can't. It's gonna take a while.
I said it first.
How do I?
No, ours already has Chromebook open.
Lipid.
That's such trash. Don't share with ours, please.
It's shareable link.
Just
so-
do everyone but Arch. That was so funny.
Alright, so now what do? I have to go here, I have to go here, Classroom. I'm gonna post the link on Classroom. Is it editable though? Editable? Can I see? Others can?
Others can edit.
It's not up yet.
Yeah.
Link sharing updated. Okay. Let's see if this works.
Okay, can I write my name?
Yeah, here?
Yeah.
Nope.
Wait, hold on. Here?
Thank you.
That's not.
Oliver and Arch. Where is it? Where is it? Oh, no, we got it. I didn't share it. Cortisol. Are you stressed out? You would not have enough stress?
Okay, thank you.
Alright.
Let's go. Get bodied, Arch.
Hey.
I'm kidding.
Alright, I heard some chairs getting pushed around here.
Let's go.
Alright, it's on there now, so you guys can. I need a student to. Alright, listen. I'm coming around to check your homework from last night, which is the first Lipids worksheet, and then I'm coming to collect your labs from the gel column lab.
I'm not even putting it for that.
Can you pass this out to make sure every single person gets one, right? Thank you. You guys, Mary is passing out your packets. That's homework tomorrow. For tomorrow. Due tomorrow.
I told you, literally no one was.
Yes. You guys, the two labs, like in detail, are gonna be on the quiz on. Is it on Tuesday or Monday? Not Wednesday. Tuesday.
I got John down.
Wait, please. Did we hand in our lab?
I'm actually going to take it. Yeah. You guys, please, can you rip off the back? Can you take off the back pages?
Okay.
Back pages? Aren't they?
Of the lab. Take off the front pages, or take off the back ones and give me the back ones.
I was like, wait, you just want the papers that you gave?
Thank you very much. Yes.
Wait, it's 2 pages? Yeah, there's 2 sets, right? It's just 2 pages?
Did we have to print out the graph? We could have just shared with you the graph and the table, right?
Yeah.
I have my table printed out for some reason. I don't know why.
Thank you. Wait. I'm okay. Should I check these? Should I collect these? I don't know if I'm gonna. I might just be checking these. I'm gonna collect the lab.
I think that's the.
Oh, wait, you're collecting the lab.
I want that. The lipid thing. I'm just gonna come around and check in a second.
So this is where you can press the button.
Wait. So the lab should go here. Yeah, 1, 2. Thank you. Lab. Thank you. Lab. Good. Lab. And then I'm gonna keep those out though. I'm gonna come check them now.
Don't dance, Stella, teacher.
Right here.
Thank you. Staple the door. Staple the door. Thank you.
Thank you.
Brother Arnold. Brother Patel, I am disappointed. Sorry, I just really have to do that. Sister Adelon, I am very disappointed.
Thank you.
What a real one.
She used like 3 separate machines. She checked it herself. About the art thing. Really? What? Carly, are they actually worth that? Oh, I want to tell you that 1/3. I want to take it as an opportunity to pass out at people. What'd you say? I can't find it. What? You should have taken it down. Oh, yeah, I should have. Should have been like,
Oh wait, are we taking that off? We're just taking off the post lab section and giving it to you?
That's what I said. Yep.
I can't read it.
No, I'm gonna come around and check that.
Oh, whoa.
Okay, here you go.
Alright, can you staple it? Why is there only one page?
Oh, wait, okay.
I should have 2 pages, right?
I don't know what a stapler is, so I use tape.
Well, who took the stapler? Oh, it's right here,
It's
Sarah.
right here, Sarah.
Oh my gosh, okay.
Got it.
Oh, thank you.
Yep.
You're supposed to hand this one to you.
Yes, and staple it. Wait, which one is that? Yeah.
Illusion.
Yes. Column chromatography. Good. Thank you. 11 o'clock tomorrow. You're coming, right?
I feel like a straw. Like driving on a road.
Alright, do you want to change it? Like, do you
Oh my god, I must have got my
find something to get back to? Find something else to do instead. And your chocolate bar.
I'm so sorry.
Well, we wanted to do the pregnancy one, and then so like we're not
Alright. Okay. What do you guys? You two are?
yeah, it's, can we like do you want
Can you guys all get your homework out from last night? The lipids worksheet. I'm coming to check that.
Oh, wow. Corticosteroid treatment or driver asthma. Chronic
Denying everyone's ideas today.
We're not getting anywhere at all. It's gonna be like color of a font. I don't know. We're gonna do it based on the top.
He doesn't believe in pregnancy hormone. She doesn't want to do LDL.
Alright, so I
Yeah. Then how did they use it to treat
Whenever we land, I repeat it.
I can't.
It's a molecule that is used to attach to
It's just a molecule. It's a molecule, yes.
My drawing is really hard.
How do you guys spell hydroxyl?
H-Y-D-R. I know there's an X and a Y and an L, but that's
I'm getting so many weird spellings of it. You guys always forget the Y. H-Y-D-R-F. And then how do you spell carbonyl? Right? How do you spell separate?
I used to do E all the time.
How do you spell separate? It's the same. Separate and separate, they're same spelling, both with an A. You learn something new every day, right guys? Stop.
I should look up how to fix my technology before. This one is the new one. We're going on. Oh, well,
Straight line. Say that.
By the way, I'm leaving early.
Okay, make sure you coordinate your groups there. So that, oh yeah, you said you were leaving another day early too.
And Monday, and I think Tuesday.
Oh yeah. Or some Tuesday.
Yeah.
You guys are leaving. What time? How early?
I think I'm leaving at 1:40.
Is anabolic steroids lipids?
I mean, that's close to that.
You're smart. Come on. No, come on. How did you get in here? You're not smart. That's good. That was easy.
Is that the one?
I don't think I answered anabolic steroids considered steroids.
It says it changes the lipid level.
It says it in the bind changes the lipid levels.
But isn't it still considered a steroid?
So it says that it has the same curve up for the four rings, but aside from that.
Oh, you do. Feel better. Okay, yeah. Tomorrow 11. And yeah. And I took your homework? Yeah.
Yes.
Alright. Thank you. Does someone have the double bond? Is that saturated or unsaturated? Does any double bond at all equal one or unsaturated? But so the difference in this question is not whether it's like has double bond or doesn't have double bond. There's one here and then there's more. So what's the difference? And then what's the so why'd you change it? So yeah, so they have one or more, they're unsaturated. Unsaturated. So then they're straight and they
For this one, what actually
it's just a molecule that combines with three fatty acids to produce lipids. That's like C1. So a molecule, yeah.
I did it. I spelled it. As Bonnie Jessica, this one was true yellow. I know, we said it all in. Because on the book, mostly it's water Jessica. So we call it water Jessica ever since. Anything he's in so then I wrote it down. So it's true yellow. So yeah. I said you're the president of this and I shared your name. No, I didn't.
We had like 5. Yeah, so you want to give
Quick little problem.
We only have English and Spanish, right? And Mexican. Oh yeah.
So you said only, but we have 3 quizzes
both
tomorrow.
have quiz, and I'm not going to get home until like 10.
I'm gonna have this quiz. Don't even count. Trial in 1994.
Oh yeah, we're leaving green to Omar. Yeah, we should. Of course we're gonna.
First try. That was Jasmine. That was not me.
So how's your studying on this one?
Alright, you guys, can you put the computers back?
Like